another short one today, just looking at another form of biotechnology, is gene therapy. So, in terms of gene therapy, the main aim of gene therapy is to treat genetic abnormalities, and that's by firstly identifying a faulty gene found within the human body and inserting healthy ones in its place. So some examples of where this is currently being applied is cystic fibrosis, sickle cell anemia, and in muscular dystrophy. So again, really simply, we have a cell with a non-functioning gene. What we want to do is we want to add DNA to this with a functional version of that lost gene. So a version of that gene that is not working, we want to replace with a healthy one and ultimately lead to cells which function normally. So in order to do this, something that's commonly used is known as a vector. And a vector is most commonly a bacterial plasmid or a viral phage. And this is used to transfer the genetic material from one cell to another. So when we're talking about gene therapy, a number of key steps here. Firstly, we need to obtain a normal copy of that human gene. So that human gene could be a whole bunch of different things, but let's take our first example, so a healthy cystic fibrosis gene. So what this will then lead to is we take that gene out using restriction enzymes and we select a vector to carry that particular gene. Now, what we do is we insert the gene into the vector, and the vector is introduced into a sample of the patient's own cells. And what this prevents is the patient from producing an immune response to the introduction of external body cells, because this vector introduces a gene into the patient's own cells. Once this is done, the patient's cells are then cultured, and when we're culturing them, we're making sure that they're amplifying that correct gene that we've introduced. The cells are then transferred back into the patient. So another representation of this, we take cells from the patient. The virus, or the vector, okay, in this case we've gone for a virus, and our last one we went for a vector, is altered so it cannot reproduce in its own way, and we insert the gene, the desirable gene, into the virus. Okay, so the virus that's been altered with the desirable gene is then mixed with the patient's cells. The cells themselves become transgenic, meaning that we've now inserted the gene or the correct functioning gene into the cell that previously wasn't working. Those altered cells are injected back into the patient's body, and that may result in the production of a desired protein or a change to gene expression.